Okay, good. One is coming. Okay. Still, let's sit here. Let's go. Okay, the first one you have to make a hypothesis, but you have to observe around yourself first. Make an observation, right? And you have to questions about all things around you. When you have a questions, you can have the hypothesis that what is the answer for that question, isn't it? And when you have a questions and have a hypothesis, what's next? You have to prove your hypothesis. By design the experiments for each hypothesis. You can have 5 hypotheses or much more, but you have to start with one by one. And then when you prove it, so you can answer that it's right or wrong. If it's right, so you can answer your question easily. But if it's wrong, what else? What next? Yeah, do it again with the next hypothesis, isn't it? So design another experiment. That's very easy until you got the result. That's what the scientist does. So look at our scientists in the old time. Have you ever seen this book before? Yeah, I swear to God that it's my first time too that I saw this when I prepared this slide. It's called Myographia. So this one is from Robert Hooke in 1665. God, that's a long time ago. So he the first one who built up the microscope, isn't it? And then he looked at the cell, which kind of cell that they saw, that he saw. The first cell. Tick, tick, tick. Huh? Cork cell. Yeah. He looked at the cork cell, just like put in the top of wine, and then he just look at it. So he said he see that a square of the box. So it's a lot of square in there. So he calls it cells. Yeah, that is the first time that cell come from. And then he did not he did not propose the cell theory. He just published that, he draw the picture of many things, many organisms in his book. And then this guy, Schwann and Schleiden, yeah, they both just study in plants and in animals. And then they wrote the email, not the email, sorry, letter to each other, and they conclude together that, here is the theory of the cell. So we call it classical cell theory that come up with these two guys. Yeah. So what is the first one? All organisms are made from cells, of course, right? Animal, plants, bacteria, whatever, made from cells. And what is the second one? Of course, it's supposed to be basic, right? We don't count electron, we don't count neutron or whatever. Just count about the cell for living organism. Yeah. And what is the last one? Yeah. Yeah, Rudolf Virchow. He is the first one who found this, but it's very late, right? 1858. He found that cell always come from cells. Before that, they believed that cell can come from anything, just like the pea or the flea, sorry, the flea can come from the dust. Yeah, they they said it's a spontaneous generation, just like, the dust can create everything, create worm, create a flea, create an insect. That's what they believed before. But Virchow, he found that it's not like that. It's always have the biogenesis. That organism must come from organism. Yeah, so that's why he create cell come from cell. Okay, but right now we have a modern cell theory that's much more deeper. That we found that DNA is passed between generations, isn't it? And the second one, every cell have the similar structure and chemical chemical or just like mechanism. If you look at the plant, they have the cellular respiration, just like in the human. So that is the kind of normal of mechanism that happen in each organism. So we are all similar, especially between species. If you have the same species, you have the same mechanism and also have the same structure. That's the thing. And after that, you can see that energy will flow from cell to cell. Isn't it? ATP can produce in mitochondria, but it will be transferred to other cells. Yeah, so energy will flow. Okay, do you have any questions up to now? Yeah, okay, we'll go further. So look at about organism. How many kind of organism we have? Just two. Unicellular and multicellular. What is the unicellular? Bacteria, protists, and algae. We call algae. Yeah, algae is just sometimes is a unicellular, not all algae. Okay, not sometimes, just some algae. Okay, but many of them are multicellular organisms also. Do you know this one? Chlorella? Did you do any lab already? Not yet. Oh, not you don't have a lab. Oh God. Okay. No. Sorry, but just look at the picture. If you look in the microscope, just pick up any of water in the pond. Ah, huh? อะไรนะคะ เนี่ยครูก็งงอยู่ว่ามีหรือไม่มีแลบไม่มีใช่ไหมอ๋อไม่มีแล้วเนาะคือจริงๆอะครูพักเบรกไป1 ปี I was sabbatically for a year. Yeah, I just did for research. So I just came now, start teaching. Today is my first class. Yeah. So I didn't know that you have a lab or not have a lab anymore. Okay. Oh, that's fine. That's why you can study in your faculty. Much more interesting, I believe. So when you look in the under the microscope, right, you can see many kind of algae. This one is a chlorella. It's a single cell that's always circle. It's full of chlorophyll in there. Yeah, because they ate some proteins also. Yeah, the algae ate some proteins, and the protein can eat some algae. They exchange like that. So if you see the chlorophyll in there, like your green, they just ate chlorella. And here is another one that you have to know. Yeast. You know yeast, right? Yeast is the only fungi that is unicellular. So other fungi are all multicellular. Okay, got it. Okay, for how about what is the multicellular organism? Animal. not only him. And then plants, and then fungi, of course. Fungi or fungi, whatever, that's the same. So look at the instrument that we used to study for the cells, right? The first one have to be microscope, of course. It's a light microscope and electron microscope. So light microscope have two kinds, compound and serial. So give me the example of the cells that have to use for the compound microscope. Yeah, of course. Is it have to die? Is a dead cell or living cell? Dead. Have to be dead. Everything that you look under compound microscope have to be dead, because you have to section in a very thin layer, isn't it? So if it's alive, you have to section it also to make it thin enough so you can see through of the cell. And how about stereo? Does it have to die? No, don't have to. Yeah, you can keep them alive. Yeah, there are clips. So if they are alive, you can clip it. Clip the wing over there. So you can see the outer layer. Just only the skin or the hair of them. Just the outer side, not inside at all. And here, electron microscope, of course, cells had to be dead, isn't it? Because they use heavy metal in the cell. They will embed it, all the heavy metal in there. So it can resonate with the instrument. So you can see the structure of the cell inside, or can see the very small thing, because the magnification is more than 100,000. See? The first one is the scanning electron microscope. When it calls scanning, it's very easy, right? Scan from outside. You can see only the outside of the cell. Yeah. But if you use the transmission electron microscope, it can see through the inside one. Yeah. So we call this like SEM and TEM. Our faculty has two. So if you want to use it, it's very cheap here. So just come here to use. We have the new version also, just got it. Yeah. And when you look at the quality of the microscope, how can you tell? You have to look at the magnification of them, right? Sometimes it can go to 1,000 for the light microscope. 1,000 doesn't mean that this eyepiece is 1,000, right? This eyepiece will be only 100, but it will multiply. Oh, sorry, this is the objective lens. Sorry, yeah, not eyepiece. This is the objective lens, will be maximum 200. Have you ever used it before? How many of you used it before? 1, 2, 3, 4, 5. Yeah, but you can imagine, right, how it works. Others, so normally they have 4 magnification. They have the eyes lens, the eye we call eyepiece, that will stick into our eyes, and they have the objective lens, so it will multiply together for the whole magnification, total magnification. For the eyepiece, it's normally only 10. But for the objective lens, it will be varied from 4, 10, 40, and then 100. So for the 4, if the objective lens is 4, so what is the total of magnification of this microscope? 40. Have to be 40, because you have to multiply with the eyepiece. That always 10 all the time. And how about the 10? Total will be 100. Yeah, easy like that. And then 40, 400. And then 100, 1,000. Yeah. And quality of microscope depend on the magnifying power and also the resolving power or resolution. Yeah. For this one means that if you put 2 things together and you have very little space, if the resolution is high enough, it can separate 2 things from each other. If the resolution is not good, so these 2 things will overlap all the time. Just like this one, this is the old picture that we got from the very old microscope. So it's very blurry, right? But right now, you can see that the cells separate from each other like this when you use the very high resolution. Yeah. And this one, you don't have to worry. I will not put in the exam. It's crazy, you know. Just know that when you have higher magnification, your NA here, it have a NA number, is here. Just like 4x is 0.1, 10 is 0.25. This one will go along with the resolution. Yeah, so it go together. It's like higher magnification, higher resolution. And here is the piece of the microscope that we call. So at the eye is always 10x that I told you is eyepiece. And then optic lens will be vary from 4 to 100. And they have the iris diaphragm that you can open the light. If you open more, the light will get through your cell easily. And the field of view will go deeper and deeper, isn't it? If you use 4x, you can see, no, if you use 40x, not 4x. If you use 40x, you can see a lot of the field, right? Many things in that picture. But if you zoom in, you can see one cell. That's very easy to explain. Do you have any questions? Go too fast? Sorry. Is that too fast? Okay. I will try to slow down if it's too fast for you, okay? But normally I cannot slow myself. You have to slow me down. Just like teacher, slow down. Okay. So the second one is centrifuge. Centrifuge is just the spindle like this. So you can separate it from the density. Normally the density will go along with the weight, right? If you have the higher weight, you will drop down easier than others. Yeah. So we have the microcentrifuge. Microcentrifuge will go up to 15,000 rounds per minute, RPM. But if you have the ultracentrifuge, it will go to very high, more than 150,000 rounds per minute. So this one we use for the small molecule, just like here. If you just break down the cell, and then you want to separate all the organelles, so you have to increase the centrifugal power. Normally, the centrifugal power will use G, but we can calculate G force into round per minute later. You don't have to do that by hand. Just go to the website that I place here. If we have this for you, you just put the number in, and they will calculate it for you. Have you ever heard about Sigma Aldrich? This one is a very big company for every kind of chemicals that we are using here. So you can, this one will assure you that it's very correct because they are very big company. It's very precise. Yeah. And here, when you use only 600G, very small, right? For 10 minutes, you can centrifuge the homogenate and nuclei from each other. And then when you increase the centrifugal number or power, it can separate mitochondria, chloroplast, lysosome, and peroxisome. And if you increase more, it will separate plasma membrane for you. And if you increase more, it can separate ribosome subunits. And the last one, of course, cytosol or cytoplasm. See, if you increase the power, so you can get the smaller of the compounds out, okay, or compound organelles, whatever. Yeah. We studied about the unicellular and multicellular already. Now we come to the animal and plant cell. So here is the table that I separated for you. What's the difference between plant and animal that you have learned on your high school already. So we will emphasize it a bit. So it's like for the plant cell, it's larger, right? Normally it's larger than animal. Animal is a very simple, very thin, and very small cell. And which one is stronger? Plant or animal? Plant, because they have cell wall. Yes. What's the compound of cell wall? Cellulose. What the cellulose is? Carbohydrate, protein, or lipid? Carbohydrate. Or carbohydrate, of course. It just have only glucose, right? In a single unit. Lie down like this, very long chain. Yeah, so it's carbohydrate. Why the buffalo or the cattle eat grasses and they get fat? Because they are eating sugar. They have a cellulose enzyme that can digest cellulose, right, into glucose. That's why they can get energy from. But we don't have that enzyme. Although you eat a lot of grasses, so you will not get fat. And here, so the animal cell does have cell wall or not? No. But we have the cell membrane, right? What is the cell membrane? What's the compound of cell membrane? Phospholipid and have inside. You know that phospholipid, but we have protein. Yeah, we have protein between that phospholipids, isn't it? And on the protein, normally what kind of compound stick on them? Carbohydrate. Great. Yeah. Normally lipid have carbohydrate stick on them. So they can communicate with other compounds outside. So we will study it later. Right. Okay, so look at the organelles. Plants have chloroplast. Animals have chloroplast or not? No. Vacuole, normally plants have a very large vacuole. Does a plant have? Oh, sorry. Plant have very large vacuole, but animal, does animal have vacuole? They used to have. Yeah, when they get born, just originate, they have a really little vacuole, and then it will disappear after that. And the tonoplast around vacuole. If the plant have vacuole, they will have tonoplast, isn't it? But the animal doesn't have tonoplast. You know that tonoplast is the cell membrane of vacuole, right? Here, tonoplast is cell membrane of vacuole. We separate the name. And the centriole, who has? Animal or plant? Animal. Plant doesn't have centriole. Okay. How about the nucleus? They both have. But normally, animal cell, we can see the nucleus in the middle of the cell. Because nucleus is very big, very huge, right? But the animal cell is very small. So when you look at the cell, it's almost nucleus inside. But for the plant, it's very big cell and have a lot of things inside. So they will put the nucleus into the edge of it. So you can see that it's normally go at the peripheral of the cell. That means it's at the edge of the cell. Very hardly to see. How about the lysosome? Only animal have. Normally plant doesn't have, but some have. They said RA. And here, storage of material in the plant normally is a starch, right? But in animal, we store it, glycogen. Okay, here is the picture from electron microscope of the animal cell and the plant cell. You can see that the animal cell have a lot of, can you see what I point out? Which is organelle? Can you guess? Huh? Oh, can you guess that which organelles that the animal cell have the most? Just look at the picture where I point mitochondria. Yeah, normally animal cells have a lot of mitochondria inside because we have to use a lot of energy all the time. That's why we have to produce much more mitochondria. And it can divide themselves all the time, right? Especially in the heart, in the liver, the cells that have to use a lot of energy. So they will divide themselves every hour. And how about the plant cell? Which organelle they have the most? Can you guess? What is this? Chloroplast. Yeah, they have a lot of chloroplast. That's why it turned green. Okay, so we finished about the plants and animal cell. Now we start with the prokaryote and eukaryotic cell. What we are? We are eukaryotic or prokaryotic? Eukaryote. We are eukaryote, of course. And the prokaryote is only in bacteria or cyanobacteria or archaea. Archaea is very is a bacteria that in history. Okay, yeah, that's a long time bacteria. This is like, oh, okay, originate in the world is archaea and then develop into bacteria. Yeah, we call it eubacteria. EU, that means real. So just like eukaryotic cell, you mean real or true. That means we have the real genes. Karyote is just like chromosome. So we have real chromosome. Okay. So look at the eukaryotic. It has protists, fungi, animal, and plant. All of them are eukaryote. Okay. So everything in here have the envelope, right? But for the prokaryote, doesn't have anything that have envelope. So organelles that have envelope, they will not have also. Just like chloroplast have 2 layers of membrane, right? Envelope mean membrane. Okay. Chloroplast have 2 layers of membrane, they don't have. Mitochondria, they don't have. Some kind like that. Or nucleus, not even nucleus. They don't have nucleus also. But they have DNA. So here, the bacteria, it's very small size. It's very small size. But eukaryote is a bigger size. The nucleus, because they don't have nucleus, right? They just try to group the DNA together. So that group of DNA, they call nucleoid. Something that like nucleus. That's what it means. Yeah. And they have only single DNA molecules. But for eukaryote, we have nucleus already. And we have the pack of DNA in the chromosome form. And for the membrane-bound organelles, so they don't have any, but they have ribosome. Of course, they have to produce protein. They still have ribosome inside. But we have all mitochondria, Golgi complex, chloroplast, and etc. that have envelope that I told you. Okay, and have more here. Just like DNA of prokaryote is naked. That means no membrane. And DNA is circular. Okay, and no intron. Do you know intron and exon? The gene that be translated to work. You know, just like you have a long DNA like this. DNA have the intron and exon part. The intron means the piece that doesn't have any gene on it. But the part that have gene on it, we call exon. Why we need intron? It protect us from misreading of the gene. It's like when you read this first gene and then you have to stop. And then you start with the next gene and then have to stop like that. But when the bacteria, they doesn't have any stop, right? They read gene like this. That's why it can mutate very easily. Here is the organelles of the prokaryote, no nucleus, right? No membrane bound, and have only 70S ribosome. Ribosome means very small, but eukaryote, yeah, have all, but the ribosome is bigger, go to 80. And the reproduction, it use binary fission, isn't it? Yeah, and have a single chromosome that called haploid. But for us, we have mitosis and meiosis, right? And the chromosome always pair that called diploid or more. And the size is very small, up to 5 micrometers. but for us, it's more than that, of course. And here, if you look at the diversity of the organism in here, we can see that the archaeobacteria is go to this one, it's unicellular, and the prokaryotic go together. And here is a eubacteria that I told you is a real bacteria that called eubacteria. So it developed into another branch, but still unicellular. But for the things that develop after that will be eukaryote. Just like Protista. Protista is still unicellular, right? But it's a eukaryote. I will give you in the exam for sure which one is unicellular, which one is multicellular, which one is pore, which one is eukaryote. It's very important to know. You have to separate it. Okay. And here, plants, animal, and fungi, they are all multicellular together and all eukaryote. Yeah. Okay, 15 minutes left. How is your head right now? Okay, your brain is almost explode. Is it okay to keep going on? Okay, so the cell composition, you have cytoplasm, right? That's the semi-liquid inside of the cell. It's not liquid. It's just like a jelly-like, you know. So it's not watery. Yeah. And the thing that have inside of the cell membrane, we call protoplasm. Can you separate? Just like nucleus, nucleolar, cytoplasm, that doesn't count membrane, so we call protoplasm. Okay. And here is the structure of cell wall. Cell wall have primary, middle, lamella, and secondary cell wall. It's very complicated. It's not very easy like that. So you can see that here is a middle lamella of them, just like the cell between cell wall. And then you can see the primary cell wall and plasma membrane. That's only some plants that have secondary cell wall, not every plant, okay? Just some plants. When you look at the real picture, you can see here is the first cell. On the top is the first cell, and here is the second cell, okay? You can see that here is a primary cell wall, okay? Outer layer, primary cell wall, and then secondary cell wall, that's very thicker. Where is the plasma membrane? It's after secondary cell wall. And this cell also, primary, secondary, that go to brown color. And then plasma membrane. Where is the middle lamella? It's between two cells. So this middle lamella is just the space between two cells. Okay. So what's the composition of primary cell wall? They have the pectin. Pectin is a sugar. Okay. So the primary, you can see that look like the pomelo. Pomelo have a very soft of the peel outside, right? That is the primary cell wall. But they contain a lot of pectin. So pectin is made them very soft. But the secondary cell wall, they have lignin inside. Lignin is a very strong of compound. If you have lignin, it's very thick and strong. So only a tree that has secondary cell wall. Okay. And this lignin gives you a brown color. Yeah. That's why secondary cell wall turn brown, because of the compound that's called lignin. The outside of the cell wall, they will normally have the Q10, right? The Q10 is the lipid, very long chain of lipid. You can see carbon 20 of carbon long, and 24 of carbon that have alcohol. Okay, that's long. So it's a wax. Very long like it will turn into wax. So we protect them from the, what is the English? What is the English? Water, 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 respiration. Yeah, this one is reduce the respiration of the plant. Okay, if it's in the drought area, it's very hot area, so it will, the respiration of them will be very fast, right? So each time of respiration, there will loss of water too. So it will reduce the respiration. So if you just put the drop of water on it, you can see that it cannot go inside. Okay. And here between, I would like you to see that the plasmodesmata, when the plants have cell wall, that's very strong like that, but they can communicate between this channel. Okay, then we have a little channel between them. We call plasmodesmata. Okay, that's why they can transfer all the compounds from cell to cell through these channels. And here is the cellulose of the primary cell wall that you can see each molecule is a glucose, right? So then we have a long chain of glucose that pull up together. So we call microfibril. Here is the microfibril of the cellulose. Okay, that have a lot of cellulose molecules in there. That's why it's so strong. And I would like you to see here for the primary and secondary of cell wall. For the primary, mostly of them is a cellulose. But for the secondary, then we have the lignin here. Okay, here is the compound of lignin. That's all between them. If you look at the chemical structure of the cellulose, mark this slide, it will be in your exam. You have to remember all kind of the chemical structure too. Okay, and here you can see glucose connect with glucose, a long chain is a cellulose, right? And here they have the word that called hemicellulose between the cellulose too. The hemicellulose is just like the cellulose, but they have a branches. See, it's not go to the long chain like this. Then we have another compound go up. So that we call branches of compound here. And the lignin is an alcohol, but they have the benzene. Here is the benzene, right? And they have other compounds that have OH group or hydroxyl group here. That make them turn to alcohol. This compound is a lignin. Okay, look at the picture. The biggest one is cellulose. Smaller green one is hemicellulose. And the brown one will be lignin. So they will wrap around together to make them very strong. You can extract the pectin from the peel. Mostly they are in citrus or pomelo, in citrus fruit or lemon, right? So when you separate it, extract them, you can make a powder. Mostly of them are the yellow one. But if you bleach them, it will turn into white. So this one can mix with the jelly. Yeah. So all kind of jelly, that's all mixed with the pectin. Okay. And the hemicellulose, you can extract them too to use as a fuel. Right? You can use as for a gas. But the lignin, you can extract it to use for the medicine. This one is a very strong agent for stop breathing. If you have a breathe, So just pour lignin on it. It will stop immediately. I think you will use it, yeah, for your patients in the future. Yeah, it's very strong, and normally it's mixed in the medicine already. The fruit that have a lot of lignin is the mangosteen peel. Mangosteen peel is very dry, right, and astringent. We call the astringent taste. Yeah, when you bite it, you can feel weird on your tongue because of the lignin that precipitate your protein. That can precipitate everything, especially protein. That's why it can stop breathing, because it precipitates the blood, right? And cashew, you know cashew? Yeah, they will bite it. The old people, they will bite it and turn the teeth into black. Yeah, that one have a lot of lignin also. In Taiwan, they still sell a lot of cashew along the road. And they have a lot of incidents that they have the mouth cancer or the tongue cancer, because that cashew precipitate the protein inside the mouth and cause cancer finally. So the right amount is good, not too much more. Okay, I think I will stop at the here. Okay, not too many slides. You can be patient, right? Now we go to the cytoskeleton. What is the compound of cytoskeleton? Yeah, what is the compound? Protein, carbohydrate, or lipid? Protein. They are all protein. Remember that. Cytoskeleton are proteins. Okay? But they make like a network like this, just like a net like. So they will have all around in the cytoplasm. Why they have to do like that? Because, yeah. Do you know? Do you know what it means? Anyone can explain in English? So they make a network because they try to control the structure of the cell, isn't it? And they try to control the movement of organism. So organism cannot move inside the cytoplasm freely, not just flow like this, but they move through this protein network that the cytoskeleton made of. So they move in the system. Everything in the cell is the system. Not move just, okay, I want to go left, I can go left. Okay. And the biggest one we call microtubule. Look at the blue one. We call microtubule. And the middle one we call intermediate filament. And the smallest one we call actin. Here, actin, or we call microfilament. So it's the same thing, okay? Microfilament, normally we call actin. It's the same word. Don't confuse. So what is the biggest one? Second. And the last one? Or actin. Perfect. Okay, now you know that they control the structure of the cell and control the movement of organelles. Okay, good. And here, I write down for you, provide structural support. It maintains the cell shape. Enable cell movement, right? So organize the cytoplasm, just like enable the organelles inside the cytoplasm, and facilitate intracellular transport, right? So it will control the cell division too. Here is the animal cell division. Okay, they will use the actin here as a contact time ring, and then they will separate the cell from each other. Okay, you can call cleavage furrow if they start to separate the cell like this. You can see the crypt here. So they call cleavage furrow. Okay. And here is the movement of the cell, right? So it will wax like this. Here is the what is that? Is a tail of the protease, right? So it can work like this to make the protease go forward. Yeah. And here is the amoeboid movement. So here are the proteins that try to control them to move forward. And this one is our muscle. Then we have the myosin and actin here that control our muscle movement. In the next topic, I will teach you of this. It's very hard topic, okay? And here amoeboid movement. You can see that the chloroplast will move inside like this, move around. So that because they have the cytoskeleton that we cannot see, but it will move only around. Okay. We call a cytoplasmic streaming. If you have the lab, you can see it clearly. Yeah, as a chain. If you have a problem with the intermediate filament, you cannot move anything also. Do you know him? Have you ever seen this movie? Do you have Netflix? Or you can type in your Google if you have a full movie in the Google. Yeah. Theory of Everything. I recommend you to watch it. Stephen Hawking, he discovered just like the theory of the universe. He is very smart, but when he just like turned up to be a rising star, he started to have the disease that called amyotrophic lateral sclerosis, or ALS. Okay, or we call Lou Gehrig disease. This one come from the name of baseball player. Yeah, he is a superstar too, but got the disease later. They all have the problem with the intermediate filament that connect to the motor neuron to the muscle. So when they don't have any neurotransmitter to them, right? So the muscle cannot work anymore. So they don't have the power of their muscle. They cannot walk or cannot speak. But it's very genius of him that he create his own computer that can translate his signal from the brain into the word on the screen. And then he create that it read out loud so he can talk with other people, just like communicate normally. So you can see him on the wheelchair and with his computer that have a speaker all the time because it here, order from here. That's very smart. So this is his book and this is his movie. You should know him. Yeah. It will be amazed how amazing he is. But the funny thing that when he got this disease, he still have a mistress. You know that? Second wife. I don't know how. Yeah, I don't know how. But his mistress is his nurse. So just watch the movie by yourself. It's very funny. Okay, I think we should stop here. And we can start later because it connect to each other. Okay, see you when? Next Thursday, isn't it? Tomorrow. Okay, right. Okay, tomorrow 8. Okay. Bye-bye. Good luck, students. You too.